Okay, so welcome back to this next video in which we are discussing phospholipase C enzymes. Okay, right, so we've just been discussing phospholipase C delta family enzymes. What we're now going to move on to is the phospholipase C epsilon family. And remember, in the phospholipase C epsilon family, there is only one member, okay, and this is just called phospholipase C epsilon. Okay, right. So phospholipase C epsilon has a very broad expression. It's found expressed in a huge number of different cells. Okay, and it has quite a different structure to the other phospholipase C enzymes that we've seen. Okay, some of the bits are going to be common to other phospholipase C enzymes that we've seen, but we're going to have 3 new domains that were certainly not in other phospholipase C enzymes that we've had a look at. Okay, so let's start by drawing out then the structure of our phospholipase C epsilon family phospholipase C enzyme. Okay, so here's the amino terminus of our phospholipase C epsilon protein, and then here is one of these new domains that we haven't seen before at all. Okay, and this is what's known as a CDC25 domain. Okay, and this is why phospholipase C epsilon is going to have a very different role to any of the other phospholipase Cs that we've seen so far, because this CDC25 domain is actually going to function as a RASGEF. Okay, so when the phospholipase C epsilon enzyme is active and its phosphoinositide phospholipase C activity is active, the CDC25 domain will also be active and it will be functioning as a RASGEF. And if you don't know what a RASGEF is, then we'll come on to it in a bit more detail in a moment. Okay, but for now I'll just put it out there for those who do know. Okay, right. Then after the CDC25 domain, what you're then going to have is the more normal domains. Okay, so here you're going to have that same old plexgen homology domain, and it's going to go back to being useless basically. It's not going to do anything in phospholipase C epsilon. It's just there. Okay, then something's different here. You do have EF hand domains, but you only have 2 this time. You have a single EF hand dimer, okay, rather than the 2 EF hand dimers. So you only have 2 EF hand domains, 1 EF hand dimer, rather than the 4 EF hand domains that we've been used to in the other phospholipase C enzymes that we've had a look at. Okay, so that's one of the big changes for phospholipase C epsilon. Then after that, of course, we have our tin barrel-like domain, which is the same as always. Okay, being made up of an X box and a Y box, and then in between the X box and Y box, again you have the XY linker, and that's performing this auto-inhibitory function where it sits within the catalytic site and inhibits it. Okay, right. Then after the Y box, you'll have the C2 domain. Okay, and again that's not doing anything in the phospholipase C epsilon, it's just there. Okay, and then after the C2 domain, you have 2 new domains that are again unique to phospholipase C epsilon. Okay, and these are going to be RAS association domains. So we've seen that the CDC25 domain down here is a RASGEF, and now we're going to see RAS association domains. Okay, so something, well, this is screaming out that this enzyme is incredibly involved with RAS. Okay, and we'll see what that involvement is in just a moment. So here is our first RAS association domain. So RA stands for RAS association domain. Okay, so RAS association domain. So this is the first RAS association domain, hence why it's called RA1. And then after the first RAS association domain, you then have another RAS association domain, which is called the RAS association domain 2, and then you finish. There's the C terminus. Okay, so I'll colour in these RAS association domains in yellow here. Okay, so here's the RAS association domain 2 and there's the RAS association domain 1. Right, okay, so normally phospholipase C epsilon is in the cytoplasm. Okay, so in order to activate the phospholipase C epsilon, you need to get it up to the plasma membrane, and you also then need to reorient it so that the tin barrel-like domain here is adjunct to the underside of the plasma membrane so that the XY linker is then repelled out. Now, when you do this, when you activate the phospholipase C epsilon enzyme, not only do you activate the hydrolysis of phosphatidylinositol 4,5-bisphosphate to diacylglycerol IP3, but you also activate the CDC25 domain. Okay, and this is going to act as a RAS-GEF. So let me now tell you what a RAS-GEF is. So let's start with this GEF. This stands for guanine nucleotide, that's the G, okay, and then the EF is for exchange factor, so it's a guanine nucleotide exchange factor, and it's going to basically exchange guanine nucleotides on the RAS proteins. So the RAS proteins are a family of monomeric G proteins that are all attached to the underside of the phospholipid bilayer. Okay, so there are 3 different genes for RAS proteins, okay, called HRAS, NRAS, and KRAS. Okay, so there's HRAS, NRAS, and KRAS. Okay, and for our purposes they all do exactly the same thing. Okay, so often they're just abbreviated down to RAS. So when I put RAS here, that means one of these. Okay, so either HRAS, NRAS, or KRAS. Okay, and it's attached to the underside of the phospholipid bilayer by a lipid moiety. And again, it's a palmitoyl group specifically that's attached to RAS, but the specifics don't really matter. The concept that you've got this lipid moiety attached to you, which is then implanting into the inner leaflet of the phospholipid bilayer, that's the important thing. Okay, so RAS is attached to the underside of the phospholipid bilayer. Now, being a monomeric G protein, all the RAS proteins can be in 2 states. They can be in the off state where they have guanosine diphosphate, GDP bound to them, and they can also be in the on state where they have guanosine triphosphate, GT bound to them. Now, normally RAS monomeric G proteins are in the off state, okay, but they can be activated to be put into the on state by RAS-GEFs. Okay, so things with CDC25 domains in them. Okay, right, so phospholipase C epsilon is a RAS-GEF. Using this CDC25 domain, it can bind to the RAS monomeric G protein, and then it can catalyze the release of the guanosine diphosphate, and then the binding of guanosine triphosphate. Okay, and then the RAS monomeric G protein will cleave away from the CDC25 domain, and it will be in the on state, and then it can have all sorts of different signaling roles. So for instance, it can participate in the RAS-RAF-MEK-ERK pathway, okay, which is extremely important in controlling cell division. Okay, so the RAS-RAF-MEK-ERK pathway, okay, in which RAS activates RAF, which then activates MEK, which then activates ERK, and ERK goes into the nucleus and then phosphorylates certain transcription factors and causes a major epigenetic shift within the cell. Okay, right. The other thing to say is that not only is phospholipase C epsilon actually capable of activating the RAS monomeric G proteins when it is activated and at the plasma membrane, okay, but it's also capable of actually being activated by monomeric G proteins. And you might think, what? How does this work? But let me just say 2 words to you: positive feedback, and I'll explain this now. Okay, so let's start off with how phospholipase C epsilon enzymes can be activated by RAS monomeric G proteins in the on state, and then we'll look at how does this all come together, how does this help in positive feedback. Okay, and we'll look again at receptor tyrosine kinases. Okay, right, so firstly, how can monomeric RAS G proteins in the on state activate a phospholipase C epsilon enzyme? Okay, well, let's just draw the monomeric G protein in the on state attached to the underside of the phospholipid bilayer here. So here's our RAS G protein with the GTP bound. Okay, so basically on-state RAS monomeric G proteins can bind to the second of the 2 RAS association domains on the phospholipase C epsilon. Okay, so this is capable of now binding to RAS association domain 2, which I'll draw here, much bigger than we ever had it before. So this is RA2 here. Okay, and here's the C-terminal of the phospholipase C epsilon enzyme. Okay, and this is going to not only recruit the enzyme to the underside of the phospholipid bilayer, but it also recruits it to the underside of the phospholipid bilayer in such a way that it orients the Tim-Barrel domain at the underside of the phospholipid bilayer, and therefore actually activates the phospholipase C epsilon enzyme. So if I just draw this, here's our RAS association domain 2 bound to our RAS monomeric G protein. Here now is our Tim-Barrel domain, which is now oriented so that it's sitting right underneath the inner leaflet of the phospholipid bilayer, and this will, through electrostatic repulsion, move that XY linker out of the catalytic site, like so. Okay, so that the catalytic site is now open and available for PIP2 molecules to stick their heads into it and get their heads chopped off to produce diacylglycerol and then IP3. Okay, so then just to complete the picture up, here is the EF hands, here is the plextrin homology domain, the pH domain, and now we've got our CDC25 domain down here, which is now active and available to turn more RAS monomeric G proteins which are in the off state into the on state. Okay, right, so by activating the phospholipase C epsilon enzyme through our on-state RAS monomeric G proteins, you activate not only the phosphoinositide phospholipase C activity over here, okay, but you also activate the RAS-GEF activity down here, and this is how it's different from all the other phospholipase Cs. So far, we've just been looking at an enzyme where its only function was to hydrolyze PIP2 down to diacylglycerol IP3. Now we're looking at an enzyme that has two functions, basically, that seem completely distinct. Okay, right. So, how does this all fit together into a pathway then? Because we've got the thing that activates the enzyme is also one of the things the enzyme produces. Okay, so basically, when you get RAS activated by other means, such as by receptor tyrosine kinases, what can then happen is the RAS can activate phospholipase C epsilon enzymes, and then the phospholipase C epsilon enzymes can positively feed back the whole pathway by producing more on-state RAS monomeric G proteins and amplifying the signal hence. Okay, so let me just remind you of how receptor tyrosine kinases can activate RAS monomeric G proteins. Okay, so again, I'll draw out my receptor tyrosine kinase dimer here. So we'll imagine once again that this is a epidermal growth factor receptor here. Here is the epidermal growth factor molecule bound to the extracellular domain of the receptor tyrosine kinase, which, if this was epidermal growth factor, would need to be epidermal growth factor receptor. Okay, they've then dimerized together. Okay, so you've got another receptor tyrosine kinase here, also with its ligand bound to it, like so. Okay, and I'll do a bit of colouring in. So here are the ligand molecules in orange. Okay, and we know that when you dimerize these two receptor tyrosine kinases together, one of the key things that they first do is phosphorylate each other on these crucial tyrosine residues of the tyrosine kinase domains, okay, and that this activates those tyrosine kinase domains, okay, so that they have much higher activity than they had previously. Okay, and following that, what they're going to do is phosphorylate a whole bunch of tyrosines on each other's intracellular aspects. Okay, so here are a bunch of tyrosines, and they're now all going to be phosphorylated to become phosphotyrosine residues. Okay, now, the way that receptor tyrosine kinase dimers, like so, with all of these phosphotyrosine residues then signal is, as we know, by signaling proteins with SH2 domains being able to bind to these phosphotyrosine residues. Okay, now we saw that phospholipase C gamma enzymes could bind to phosphotyrosine residues through those NSH2 domains. However, there's another protein that I want to now introduce you to which also has an SH2 domain, which is being shown here. Okay, so this green portion is the SH2 domain of this protein, and this protein is what's known as GRB2. Okay, so this is representing GRB2, and this stands for growth factor receptor bound protein 2. Okay, so growth factor is the G, and then you've got receptor bound is the RB, okay, and then it's growth factor receptor bound protein 2, okay, or just GRB2 for short. Right, and that is supposed to represent the Sark homology 2 domain of growth factor receptor bound protein 2. Then what's going to happen is once you've got GRB2 mounted onto one of these phosphotyrosine residues, another protein, which is also a RAS-GEF, comes and binds to the GRB2, which is the SOS protein, okay? And the SOS protein also has a CDC25 domain and is capable of RAS-GEF activity, okay? So it's capable of converting off-RAS monomeric G proteins. And understand, before you've ever stimulated anything, the RAS monomeric G protein should be in the off state. Okay, so SOS is now capable of activating RAS monomeric G proteins. It's capable of turning RAS GDP, the off-state RAS monomeric G protein, into RAS GTP. Okay, so this is one of the ways that you can activate RAS monomeric G proteins via receptor tyrosine kinases, and then the RAS GTP can then go off and signal through the RAS-RAF-MEK pathway. Okay, now one of the ways of positively feeding back this signaling is for the RAS monomeric G proteins that are in the on state to also activate a few phospholipase C epsilon enzymes. Okay, and then what that will do is recruit the phospholipase C epsilon enzyme to the plasma membrane, and that will have two effects. Firstly, it means that now the CDC25 domain is up nearby RAS monomeric G proteins, which are attached to the underside of the cell membrane, so they can start now converting RAS monomeric G proteins from the off state to the on state, thereby producing more and more RAS monomeric G proteins in the on state, so amplifying the signal. In addition, the phosphoinositide-specific phospholipase C activity can also help the phospholipase C gamma enzymes, which have also been activated by receptor tyrosine kinases. So this is another way that you can activate phosphoinositide-specific phospholipase C activity via receptor tyrosine kinases, basically. Okay, and therefore from that you'll produce IP3 and DAG. Okay, right, so that's one way to activate the phospholipase C epsilon through the RAS monomeric G protein, and it serves to amplify the amount of RAS monomeric G proteins in the on state. Okay, there is, however, another way which I now want to mention, okay, which is that you can also do it for G protein coupled receptors, okay, but through very rare G protein coupled receptors. The G protein coupled receptors that we are about to discuss are G12/13 coupled G protein coupled receptors, okay, which are coupled to heterotrimeric G proteins with alpha 12 or alpha 13 subunits. Okay, right, so firstly let me discuss some examples of G12/13 coupled receptors, because they're rarer than you would think. Okay, so here is the cell membrane, okay, and we'll draw our G protein coupled receptor with its characteristic 7 membrane spanning alpha helices. Okay, so a class of a class of G protein coupled receptors, which are all G12/13 coupled receptors, is the thrombin receptors. Okay, which are the receptors for the protease enzyme thrombin, which is activated within the coagulation cascade and activates fibrinogen into fibrin monomers. Okay, so the thrombin receptors are what we're going to discuss. Okay, so the thrombin receptors have another name. They're also called the protease activated receptors, and they have a very curious mechanism of activation, which I'm not going to be able to show with this picture. I'm going to have to extend that N-terminal domain up a little bit. Just realized that. Okay, so the protease activated receptors, which are also usually abbreviated down to the PARs. Now, you have 3 PARs, which are called PAR1, PAR3, and PAR4. So similar to the phospholipase C delta enzymes, there is no PAR2. Okay, right. Now they have a very curious mechanism of activation. Basically, what happens is the thrombin enzyme, okay, which I'll draw over here. So this is representing the thrombin enzyme. Okay, so I'll colour in thrombin in orange here. Okay, so that's thrombin. It's going to cut the N-terminal region of the G protein coupled receptor, of this protease activated receptor. So this is now a PAR receptor here. Okay, and basically what's going to happen is thrombin is going to cut the N-terminal domain of the PAR receptor. Okay, and what that will now expose is a new portion of the N-terminal domain here, this portion that I've now highlighted in orange. And basically this new portion that you've exposed, that is now the ligand for the G protein coupled receptor. It's what's known as a tethered ligand, okay, because it's effectively already attached to the G protein coupled receptor. So you cut off the top of the amino terminal portion, that bit goes off. This newly exposed portion here in orange then goes down and binds to the lower portions of the G protein coupled receptor, causes a conformational change in the G protein coupled receptor, which then activates it. Okay, and this G protein coupled receptor, once activated, is then going to work through G12/13 heterotrimeric G proteins. Okay, so let me just draw a G12/13 heterotrimeric G protein. So basically, G12/13 heterotrimeric G proteins have as their alpha subunit either the alpha 12 subunit or the alpha 13 subunit. So there are two alpha subunits of heterotrimeric G proteins known as alpha 12 and alpha 13. And if you are a G12/13 heterotrimeric G protein, all that means is that the alpha subunit that you contain is either alpha 12 or alpha 13. Okay, and then once again, we're not going to dwell too much on what the beta and the gamma subunit are. Okay, we'll just put beta and gamma there. Okay, so this is the off heterotrimeric G protein here. What can now happen is when the PAR receptor has been activated by thrombin, cutting that more extreme portion of the amino terminus off and creating this tethered ligand, which then activates the transmembrane domains of the G protein coupled receptor, then what's going to happen is the G protein coupled receptor is going to bind these G12/13 heterotrimeric G proteins, and that's going to catalyze the release of the guanosine diphosphate from the alpha 12/13 subunit, and then GTP will come in as usual, and then of course the alpha 12/13 subunit will split apart from the beta gamma complex, and you'll then have the alpha 12/13 subunit and then the beta gamma complex. Okay, so what does the on-state alpha 12/13 subunit of a heterotrimeric G protein do then? Okay, so let's just draw this here. So here is our alpha 12/13 subunit here. Now in the on state, so it has GTP bound to it. What does it now do? Well, it recruits a protein from the cytoplasm, basically. So a protein that was previously in the cytoplasm is now going to come and bind up to this alpha 12/13 protein here. Okay, and this protein is what's known as P115, a Rho-Jeff. Okay, so this, as its name suggests, is a Rho guanine nucleotide exchange factor, and Rho is another example of a monomeric G protein. Okay, so let me colour this in. So here is the alpha 12/13 alpha subunit of the heterotrimeric G protein. Here is our P115 Rho-Jeff protein, which came from the cytoplasm and has now bound to the on-state alpha 12/13 subunit, which is attached to the underside of the cell membrane by the lipid moiety there. Okay, now the P115 Rho-Jeff is bound to the alpha 12/13. What can happen is it's going to gain its activity, okay, so it's going to be activated, and once it's activated, it's now going to start activating monomeric G proteins called Rho monomeric G proteins. Now, Rho monomeric G proteins are usually in the cytoplasm when they're inactive, i.e. when they've got guanosine diphosphate bound to them, and then when they've got GTP bound to them, they then attach to the cell membrane like the RAC2 monomeric G proteins that we saw earlier. Okay, so this is a Rho monomeric G protein. Now, when we say it's a Rho monomeric G protein, we mean it's one of three G proteins. There is Rho A, there is row B, and there is row C. But for our purposes, they all do exactly the same thing, so we will just put row there, and it means one of row A, row B, row C. Okay, and they're all monomeric G proteins. Okay, so row monomeric G proteins in the inactive state here in purple will be within the cytoplasm. Okay, and what can happen now is once the P15 ROGEF is now attached to the alpha-12/13 subunit and is therefore active, it can now activate row GDP here to row GTP. Okay, so now what you're going to get is row in the on state where it's got GTP bound, and this row in the on state with GTP bound is then going to attach to the underside of the phospholipid bilayer. Okay, and then when you've got row GTP bound to the underside of the phospholipid bilayer, so let me just show this. So here we have our row here in the on state with GTP bound. This can now do the same thing as RAS monomeric G proteins could do to the phospholipase C Epsilon enzyme. Okay, i.e. they can bind to that RAS association domain 2 here, just like RAS monomeric G proteins could, and therefore they can recruit phospholipase C Epsilon enzymes to the underside of the plasma membrane, and they also orient them in exactly the right way to have it such that the tin barrel-like domain is going to be adjunct to the underside of the phospholipid bilayer, and so that the XY linker will be electrostatically repelled out of the catalytic site, and therefore row GTP can now activate phospholipase C Epsilon, and therefore you can, through this mechanism, activate RAS monomeric G proteins and also the production of IP3 and diacylglycerol, since phospholipase C Epsilon does both of those activities. Okay, right, so that concludes our discussion of phospholipase C Epsilon. Okay, in the next video what we'll turn our attention to is phospholipase C Zeta, which is involved in fertilization.